Okay. You were here during last class. Although I asked you and none of you answered my question, but we were here that how the nascent chain, here you can see the RNA is being synthesized here, how the next nucleotide is coming, that we were discussing. And now in our cell, in eukaryotic cell, there are some differences. For example, overall idea about template strand, coding, but in the nascent RNA, this is the 5' end, right? And always it should go towards 3' end. So at one modified nucleotide should be attached. That is called 7-methyl guanosine. That is fixed. For every mRNA, the modified nucleotide. Why modified? This is little different from guanosine or G residue. In DNA, we have A, T, G, C, this G. But this is little different from the normal G. This is 7-methyl. Every mRNA at their 5' end, they acquire 7mg. That is also called 5' capping, 5' cap. And the RNA synthesis is going on, but where it will be stopped? That's the question, right? So synthesis is going on, some sequence are present there. So for example, this sequence, you don't need to remember this sequence. What you should understand is that we have some specific sequence that helps to recruit some ribonuclease. So tell me, what will be the function of ribonuclease? Last time I discussed that in biological nomenclature, how we denote different types of enzymes. So what function of ribonuclease? What do you think? Any one of you? Just try that. What could be the is? So you, like many of you, you are attending classes, right, regularly. So what do you, what function of nucleus? You don't know. See, asking question, people are leaving, but that's not the solution, right? So this is like, I'm just trying to know whether you can, like, getting some of the point or not at all, right? That's why. So this means any biological enzyme, like enzyme present in our body, the last three letter is AAC, like DNA polymerase, RNA polymerase. And DNA is enzyme which digest or which cleave DNAs. Enzyme that cleave nucleic acid. It can be RNA, it can be DNA, right? So ribonuclease means enzyme that cleave ribonucleic acid, that cleave RNA. That's all. Okay. So you want me to not asking question, right? So uh, this ribonuclease is being recruited by this sequence, and then it will make a cut here. So as a result of that, now the synthesis of mRNA is right here. And in case of eukaryote, particularly in case of like as I told you, not in bacteria but in eukaryotic system, at the 3' end, one poly A tail. So that means multiple A residue. This is almost approximately 200 A residues should be attached at the 3' end, and a enzyme called poly A polymerase adds this poly A tail at the 3' end, And on top of that, I would mention that means this poly A tail in the coding stand you don't have A. This is template dependent, independent, because when this ribonuclease cut this mRNA already, and then this is in a sequence independent manner, like poly A polymerase, the enzyme name poly, that means it can only polymerize adenosine residue or A residue, and then you will get something like this. This is our mRNA, which is little bit is the eukaryotic mRNA, but it has some function. It increases the stability of mRNA. Why it's required? Because in eukaryotic system, multiple RNA, mRNA, they need in cell for longer time. So if you can add some additional stability, so that it will be more, I would say it will be better for us. So as a result of that, I add 5 prime cap and poly A tail at the 3 prime end, you will get this more stable mRNA. Difference there, if you compare this RNA or mRNA in case of eukaryote and in case of prokaryote. So I told you before, in particularly in human, only around 1.5% of DNA is being transcribed into RNA. But here you will get something new, that after transcription, so for example, this is the product, this is the mRNA, okay? And this is 5 prime and this is 3 prime end. Even after some of the portion, here in this case you can see this one and this one, those portions are called intron. This is called intron. That means intervening sequence here, and these are exon. So intron, they should be removed from the mRNA. So then you will get the mature RNA. So this is mature mRNA, or also we can say processed mRNA, where you have 5 prime cap polytail at the 3 prime. In addition to that, those introns are already removed. In order to synthesize protein, this mature used as template. So just try to understand that whatever we discussed during last class, around 1.5% of our DNA is being transcribed. But here we are discussing that even after transcription, some of the portions are not required in case of eukaryotic system. Those are intron, and which should be removed by some enzymatic machinery. I am not going that else. And now for this work in 1989, Tomczak, so he got Nobel Prize, because this work is very important to understand that how particularly in eukaryotic system, how the processing of mRNA is involved. And also one important thing is, even I told you, like I believe during last class, that although we have around 20,000 protein coding genes in our genome, 20,000 protein coding genes, but we have more than 1 lakhs of protein. So that means you have less number of genes, but more number of proteins. But now I just told you before, RNA will make or will be used as template to make protein via translation. Then how this number disparity coming, that you have less number of genes but more number of proteins? So can you try, like, any guess how it can be done? Whatever I have discussed based on, so can you try to, this is again some question where you might not know the answer, but you can try actually whatever discussed during last slide, whatever I have discussed. So in mRNA, whatever I was talking about, so someone that voice is breaking loud actually, but you guys are not talking, so to whom I will ask actually. I'm using my LAN network, but still it's. So what about most of you? So like you cannot hear properly? Not right, just you can unmute yourself and you can tell that if you.
Yes, sir. There is some. There is frequently your voice is breaking.
Okay, that's why I'm just, I at least I should know, because I can try to change the network. And also I can go slowly little bit, it can also improve. So whatever I was saying, that say for example in this mRNA you have two introns. Here you can intron 1 and this is intron 2. Now at one point of time both the intron can be read and it will give you one mature mRNA that will give you a protein. Similarly, another case might be that one of the intron is removed, but other ones stay there. So this is called alternate splicing. So that will give you different product. In this schematic diagram, I am showing you this mRNA with just two introns, intron 1, intron 2. In reality, you might have many more introns, and whatever I discussed, that alternate splicing, that means sometime this intron being removed, sometime the another one is being removed. On the basis of that, you can get multiple proteins from one protein. That's why, although we have just 20,000 protein coding genes, but we have more than 1 lakh different types of protein. So that is also, can be again this term check discovery that also highlight this insight. Now, transcription at a glance. So whatever I just told you, that this is bacteria, you can see, and this is eukaryotic cell. Say for example, in our some human cell, for example. In eukaryotic cell, we have nucleus. So this is the nucleus, and inside the nucleus you have DNA. But in case of bacteria, nucleus DNA is present here, just one DNA molecule in case of most bacteria, that is circular DNA. After transcription, so here you can see transcript or mRNA, and it has 5' cap, and those are the intron. Those are the intron. So introns should be removed. And as I told you that poly-A tail should be added, that is also being added here. This is poly-A tail and this is called mature mRNA. This processing step is completely absent in bacteria. And now complexity in our system is this mature mRNA should be exported from nucleus to cytoplasm. So you can see in the nucleus, you have some pore. This is another pore. This is another pore. So through this pore, this mature mRNA should come outside. Do you know why it should come outside of the nucleus? What's the necessity? Because in cytoplasm only have ribosome, and ribosome translate the protein. So as a result of that, this mRNA should be exported from cytoplasm, and after that, translation will be started by ribosome. This is ribosome. But this is absent in bacteria. The good thing is, in case of bacteria, transcription and translation, they are together. Means here the transcription is going on, and here this is the initial part of the mRNA 5' end, and while the transcription is not yet completed, even before that translation started. So as a result of that, transcription and translation, they are coupled process in can happen at the same time. But in case of eukaryotic system, first you need to make your mRNA, then it should be processed so that you will get mRNA, and then it should be exported out of the nucleus, and then it will be translated by ribosome. That's all. I will be happy to discuss if you have any question, although you don't have, but I'm just, whatever I do for every class, I'm telling you. So if you have any query, you can ask me. Otherwise, I will go to the next slide. So now, regulation of gene expression, whatever I just, I introduced at the beginning of today's class, that now we need to that how this regulation happened. Like the question is same here, like each cell contains all the genetic for growth and development, but some of these genes are expressed all the time and other genes are not expressed all the time. They are switched on and off at need. So, this is my habit to discuss actually. That's why I'm not myself asking or try to discuss something. So if you see the second statement here, some of these genes are all the time. So based on your current knowledge, whatever you have, learned a little bit in science or living system, can you any example of some gene that should be expressed all the time? All the time it should make the product. That means product means here protein or any. So any example can you provide that particular gene should be expressed? Expressed means it will always constantly it will make it be transcribed and it will make protein. Any idea? Just from your common sense, try to answer.
So the protein that gives blood group.
Okay, I will remember that protein that gives you blood group. So the thing is, protein that gives to blood group, that you can express that protein, and that those proteins are actually present on the surface of blood cells. Then you don't need to make all the time. Okay. Again, like those, particularly those proteins should be expressed on blood cells only. But like one student already mentioned, actually, that is correct. That's why we are discussing, right? You can sometime, if I don't notice here, in the screen, so then it will be going into chat box, so I cannot see at that time. So some student mentioned here that enzymes which are responsible in are actually correct. In a living system, all cells are like they are living until this respiration process is going properly. So you have to have energy. You make energy in all the cells which are live, right? So as a result of that, definitely those enzymes should be like all the time they should be expressed and housekeeping genes. So in order to those genes should be expressed all the time so that you will get the product always, and those are the an example. I was expecting that someone of you might say that RNA polymerase, yes, because RNA polymerase is same enzyme, right? In all cells, wherever the transcription is going on. Say in eye cell, they are specialized for some particular function, and liver cell, they are also specialized for particular function. But whatever the whatever the specialized protein they need to make, but they need RNA polymerase, right? Without RNA polymerase, they will not get the mRNA. But mRNA polymerase will, make this mRNA from DNA or from the gene. So again, this RNA polymerase is an example of those should be there all the time. All genes are there, but they should be expressed. They should make RNA from those genes, and finally it will make protein can work constantly, right? So about, say, for example, I told you we have 20,000 genes. You can expect that around 50% of those are actually housekeeping genes. They are kind of active all the time. They are making some product which are absolutely necessary for our growth and development for all the time. And rest of the portion are called like tissue specific gene. They are in some cells, some particular type of cells, they are active, and in some other cells, they might not be active. Again, in a particular type of cell, they might be active because of some influence that for some condition, for some situation, they are turned off, they are not active. So that way, this complex system, I would say, is built. This is because of long years of, but this is the case what we have now. Now, uh, here, I'm giving you a simplest example. Very slowly, I will go through it. Try to concentrate, and because of discovering like lac operon, few terminology will learn new. I will explain all of them, but you should concentrate here. So because of the discovery is lac operon, French scientist Jacob and Monod, they got Nobel Prize because that is the first time they tried to explain how that means expression means from gene to RNA and RNA to protein, everything together we say gene expression. So how expression is controlled, or how, or what is the regulation? How the gene expression is regulated, that is the question. And they address this example, lac operon. So this is present in bacterial system most of the time. All this biological discovery happen in bacteria, particularly in E. coli. This is bacteria. Almost all of you heard about this name. E. coli is a modern bacteria, which is like there are many laboratory strains. So they are non-pathogenic. That means you can freely work in the lab. And the the genetic information of E. coli is known for a long time. So that helped scientists to manipulate E. coli when, as and when required. That's another advantage of using E. coli. Now, if I, like, as I told you already, that like few terminologies present here. So lac, this lac is short form of lactose. So in this case, we are only telling that lac. So now, operon, this is again another new term, operon. So in Opera also multiple player, like they are performing, not one, many of them they are working together. So operon, so cluster of genes related by function and regulated by single promoter and transcribed into mRNA, that is called operon. Like individually, all of you what is genes that you know. But now, for example, this is lac operon. Lac means lactose. So the function of this whole system, whatever we are lac operon, is to metabolize lactose, right? To break down lactose. Do you know what is lactose and where lactose is predominantly present? So what is lactose? This very simple question. What is lactose?
Sir, it's a sugar present in milk.
Yeah. So lactose is sugar, like it's glucose and galactose. They are linked by beta-1,4 glycosidic linkage. You are absolutely correct. The predominant source is so, but lactose, in order to metabolize in our body, lactose need to be broken down into glucose and galactose. Okay, and some special enzyme to do that. But again, enzymes are protein. So as a result of that, in order to break down lactose, or I would say in order to metabolize lactose, what you need? You need to make that particular enzyme. How you will make it? First, you need to have the mRNA. So that means the transcription need to be started first, and then the translation, and we get your product, that is the particular enzyme which will break lactose, and it will make glucose and galactose, right? So it's true for bacteria also. So in this case, I will concentrate on bacterial system, how it has been discovered by those scientists. So now, a single promoter. Last class I discussed that in the upstream of gene, you have promoter sequence that recruit RNA polymerase so that transcription can be started. But in this case, if you see the lac operon, you have three different genes which are color coded: lac Z, lac Y, and lac A. Those three genes. But see one interesting point, that all those three genes, they don't have their own promoter. They have just one promoter here. That means all three genes, they are under same promoter. So as a result of that, if a RNA polymerase molecule bind this promoter and then it moves to this direction, that means all these three genes should be transcribed at the same time, right? So that is, now I believe I explain all this word cluster of genes. LAC Y, LAC A, this is cluster of genes related by function. What is the function? The function is to metabolize lactose, regulated by a single promoter that I have already explained, and transcribed into one mRNA that you can understand, because if one RNA polymerase is here, and when it will transcribe, you will get just one mRNA, but that contains information to make product, to make protein, lac C, lac Y, as well as lac A, right? So in this table, I have listed what are the products: lac Z that make beta. This is the enzyme which cleaves the beta-1,4 glycosidic linkage. So breaks lactose, glucose, and galactose. Now, lac Y, another enzyme it makes, that is lactose permease. The lac A is very complicated name. You don't need to remember this. Lac A is also important, but without knowing that, still we will be able to understand the process, right? So you can ignore lac A for the time being. That's not a problem. So from the name itself, you can get what's the function of lactose permease. Imports lactose molecule into the bacterial cell. Bacteria, they are unicellular organism. That means one bacteria means just one cell. They are just moving around in some liquid culture broth or media when you are growing in lab. So lactose permease, its function is to bring lactose molecule from outside to inside. That means from the media to inside. That is what imports lactose into the bacterial cell. So first you need to import and then you can break down it using so that you will get energy from glucose molecule, right? So that's the idea of this lac operon, and you can see when this promoter will be recognized by RNA polymerase, and then if RNA polymerase move in this direction, then what it will make? It will make a long RNA that contains the information of all these three genes like Z, like Y, and like A. You can see these three different segments is color coding. And now all these particular things will be translated and it will make like it will make three different product. This is beta-galactosidase, and this is lactose permease, and this is another one. So you need to remember that. So as a result of that, if this is the case, then lactose will be properly metabolized. We would like to see how it is being regulated. Now, another complexity is coming here that in normal conditions, like when the bacteria is growing in some media means in lab, we grow bacterial cell in some liquid broth, liquid culture. Some conical flux, we have some special food for bacteria, but that is in liquid condition. And bacteria, they are growing at media. So now, if you have glucose in the media, then bacteria, they don't work to use lactose. So why they will understand or how they will understand that glucose is present here? As a result of that, I don't use lactose. In that complexity, I'm not going because that is further like much more complex scenario. So at this level, it's better not to go into that details. Just try to understand in this way that glucose is already monosaccharide. So bacteria, they will be happy to take glucose so that directly they will get the energy. But if glucose is not present and lactose is there only as their major carbon source, then what they will do? They need to metabolize lactose for their own survival, right? So as a result of that, during that time, they need to turn on the lab. Why? They have to make this beta-galactosidase enzyme, which is coming from LAC-Z gene, which will break lactose, right? So that they will get glucose and they can survive there. So that is the idea, overall idea. Now, since glucose is present normally, then all the time this operon will be negatively regulated. Negatively regulated means always it is turned off. You don't need to product out of these genes because glucose is present there, right? Only when bacteria will sense that no glucose, so it will turn on its own system, this lac operon, so that it can still survive in absence of glucose. That time it will use lactose, right? So now, if you see here, how this operon is negatively regulated all the time. That is what I'm trying to explain now. So another new term here, and then I believe no more new term. So lag repressor. This is means if I extend this lag operon repressor. This is also a protein. This is also a protein. So if you can remember, I told you that in a particular gene, you have a promoter. In case of if the gene is starting from this side, say +1 side, I told you that in a gene sequence, A, T, G, C, +1, +2, plus is we are naming, and in the upstream, -1, -2, -3, those are the nucleotide numbering. So around -10 position, that is the promoter in case. So, but in between this promoter and the starting of a gene, that portion is called lag operator. So this is promoter and this is lag operator. Then you have 3 consecutive genes, lag Z, lag Y, and lag A. Now, product, this protein called lag repressor, it is always bound to lag operator site. So now you see, just physically, just try to understand from the figure itself. If this protein lag repressor, it is tightly bound to lag operator. Now, what will be the functional outcome of RNA polymerase? It cannot move in this direction. It can't move in this direction. Why? Because lag repressor is obstructing this RNA polymerase to move in this direction. So as a result of no transcription, that means this operon is now turned off. Right? So as a result of that, lag repressor negatively regulate the lag operon. So it will attach to the lag operator. Therefore, the RNA polymerase cannot transcribe all 3 genes. That is the normal. But what happened when you don't have glucose in the media, in the environment, then you have to metabolize lactose. And now, if you have just few molecules of lactose, like that lactose can be lag repressor. So lactose here, if I tell you more correct way, this is not lactose, this is the racemic form of lactose. Lactose here. This is very similar to lactose. This is called racemic form of lactose. Little bit difference in bonding here. So now this allolactose bind to lag repressor. So whenever this allolactose bind to lag repressor, then quickly this lag repressor change its conformation. See, lag repressor now looks different. So this is because of this allolactose binding with the lag repressor. So the cannot know that repressor cannot bind to the operator. So as a result of that, it will be released. So it bind the operator anymore. So it will fall apart. So then the RNA polymerase can turn on, it can make the mRNA this change. So as a result of that, it will be turned on. So this is the mechanism how bacteria, they control the lag operon in presence of lactose or glucose, whatever you say. So now if I ask you, so what do you think that this lag repressor and the beta galactosidase, right? Beta galactosidase. So that means so this lag repressor and the beta galactosidase. So which of, like, out of these two, which one is and which one is like turn on and turn off at requirement? So what do you think, that both are turn on, turn off, or one of them is time it is getting expressed? Deep. So sometime you, like, what do you think, that you can unmute yourself, no problem. So I asked that we have two types of proteins here. One is lac repressor and the other one is beta galactosidase. So out of this, which one do you think that should be constantly turned on, like always there in the cell, and which one sometime present and sometime absent? That is the question. Anyone else?
So probably beta galactosidase, because lac repressor is not necessary if we do not have lactose. So the case you said, there's only glucose.
Okay. Okay, I'm remembering. And then someone else. So what is your name? Preet Kumar. Sometime you also try to answer my question. Preet Kumar, yeah.
Sir, I think it's the repressor because we need to keep it switched off in normal cases, since glucose is the primary source.
Yeah, so if the glucose is the primary source, that means the lac operon should be turned off. Like, in order to keep the lac operon turned off, then you need what? All the time you need lac repressor.
Yes.
Right? So as a result of that, if you see the scenario again here, so in normal time, I'm expecting that in the media you have, in that case, this gene is not, you don't need to activate this gene. That means if it is off, that is good. So as a result of that, lac repressor is, and it will tightly hold the lac operator so that in normal condition, the RNA polymerase cannot move in this direction. So the gene is off. But only the situation will arrive. That means no glucose. So the some other regulation there already, like, a lot of there, we remove that portion. So the system will understand that no glucose. So now only we have lactose. So as a result of that, during that time, genes need to be, activated so that it should be expressed. So during that time, you have to remove the lag repressor. That means lag repressor can be present in the cell. It is making, like, always there inside the cell. But this allolactose that will bind to lag repressor and it will displace the lag operator. So as a result of that, the gene transcription will take place. So here in this case, at the beginning, I put this thing in, but this one, all of you are aware that not an AND gate. So if you put there, like here in this case, presence means I'm putting 1 and absence means 0, right? So here, if you put here 1, right? So 1 should be 0 here and 0, then finally 0. So this portion, all of you know, right? The not an AND gate and their conversion. So that you see here, if glucose is present, 1 being present, and lactose is absent, then the lac operon, the final outcome here should be 0, gate system. So the lac operon should be turned off because bacteria, they will love to take glucose as their major energy source. Now both are present. Still glucose will be utilized by bacteria. So still the lac operon is off. And when glucose is absent and lactose is present, only you have here 0 and here 1, then only you will finally will get 1. So as a result of that, in this scenario, your lactose is on. So it must utilize lactose for its own survival. and no glucose, no lactose, nothing there. So no, right. So what it will use? So it cannot neither use glucose or nor lactose. They are not present here. So this is how a small, very, like, although while we are discussing this is very complex, but I would say this is very simple thing. Just we were discussing about uh, the metabolism of lactose or how it is being regulated. Forget about how our heart cell, our liver cell, and all those things are being regulated. Not small genes should be regulated. Multiple genes, they are cross-talking with each other. And the final outcome is we have specialized cell, specialized organ, they are providing different function to us. But in case of bacteria, they are just single cell. They don't have specialized, organ, specialized system, right? No question of that. They are unicellular, one cell. But still, this is that much of level of complexity there. But it's just, an example that is, I would say, good enough for our discussion. So that way, uh, like whatever we discuss all about transcription at the very basic level. Now we will discuss about the next step, translation. Right. This is also very interesting, but I skip some of those portions because like I don't want that so that you don't need to remember a lot of things, but you should get a little bit of idea. So first of all, tell me, just no problem. So first, tell me that our major goal was to